Okay,
Yes.
great. So this is going to be very brief since we're running close on time, but we wanted to just give people a heads up on a biomonitoring program that exists. So tying in the human health side of this permafrost and mercury conversation. I'm providing a lot of links in this talk so you can learn more, but just I'll cover things pretty briefly. So the Alaska Division of Public Health in the Environmental Public Health Program, we have a statewide hair mercury biomonitoring program. And this program began in 2002 with really the primary goal to collect information about exposures to mercury among most specifically women of childbearing age in Alaska, but we do have some samples from men as well as some samples from women outside the childbearing years. This test is free, confidential. What happens is a participant will have a hair sample taken, and either they send it in or the lab sends it, or the clinic sends it into the Alaska Public Health Lab. They run the sample, and the Environmental Public Health Program gets all the results. And we maintain a database of all the tests in the state. It's a convenient sample in that really we're not getting, a randomized sample from the entire state. Really, it's clinics that know about the program and send out samples, or if we go to environmental focused conferences, we have a booth and we're taking samples there. So really just taking samples where we can. We do have some targeted data. We've had some data specific to questions related to proposed development projects, also some other research projects where we've targeted some specific areas in the state. You can check out our website for more details on that piece. So the data we have, as Bob Gerlach mentioned, our data helps support those Alaska-specific fish consumption guidelines. And you can see more about that on the eatfish.alaska.gov website. But Dr. Gerlach did a great summary of that information. And then just real quickly, what data we do have from this program from 2002 to 2017, we've got almost 2,000 samples sent in. Our range is actually from 0, so we've got a few infant samples to 90 years of age. And the range of results is 0.02 parts per million to 16.05 parts per million. Our follow-up level is 5 parts per million. So I put a little representation of a histogram on the right side of the screen that really just shows where the bulk of our results lie, and that's really pretty far below our follow-up level. The average of the samples from 2002 to 2017 is 0.8, and our median is 0.5. Geographically, we have seen higher levels of hair mercury in rural areas, more from areas with high marine mammal consumption, particularly Southwest region and the Gulf Coast. We don't have as many samples from North Slope Borough, so it's hard to know what the North Slope really looks like. But that is the very quick summary of the information we do have. Again, much more information will be provided on our website, which I'll have the link again for on this next slide. So just our program website. We've got a brochure if you are interested in learning more about the hair mercury biomonitoring program, how samples are taken, and then just some additional information related to fish consumption and mercury. The 2012 EPI Bulletin is on our website, and that really summarizes all the available data up to 2012. But do stay tuned. We are hoping to update this bulletin with more current data. And then I just put information for our program if you have any questions on this topic, as well as my own contact information if you want a direct person in an email. So that's super quick, but information to ponder. Thanks.
Great, thank you, Sarah. Sarah Bowden, could you give us some advice about timing? We're coming to the top of the hour, but if we spill over, are we okay with that?
Yes, you are.
Okay. Well, let me throw it open for questions then for any of our speakers. Go ahead.
This is Jim McClellan. One quick question overall. Are these 3 presentations going to be available to look back at?
So yes, they are. I'm subbing for somebody today, and she is a much better administrator than I am. I forgot to record this meeting, so I remembered it at Sarah's presentation, and we have hers. But the other 2 were not recorded. However, the presentations themselves are online, and my deep apologies for neglecting to hit the record button.
So we'll have the slides then, Sarah, is that what you're saying?
Yes, you will. And I just need to work with Paul to get his slides up. I'm sorry. Yeah, Paul, Bob's are up, and I'll get Paul's up this afternoon. And Sarah, you know how to post yours, so if you could post yours, that would be good.
Okay, great.
Can I ask a question?
Please.
I thought this was really interesting. Thank you to all the speakers. And I have a specific one for Paul. I'm wondering about cryoturbation and how that might impact mercury content, because you were mentioning the time thing.
It's a good question.
That will mess it up a little, so I'm curious.
It does mess it up, yes. We had one core from Slope Mountain that was obviously cryoturbated. And it didn't follow any of the patterns that we saw, obviously because it was all mixed up. But you know, we didn't see the trends with depth in the Slope Mountain core. The other thing that complicates things are ice wedges, and if we run across core with a high density of ice, that again causes problems in terms of analysis. And that again is why we tried to get so many cores of different variability, because we didn't want to hang our hat on one core that might not be representative of the entire northern hemisphere. So yeah, I imagine if we keep collecting cores, we're going to run into that again with a Slope Mountain type core with cryoturbation.
This is Jim McClellan. I've got another question. That's all right. For Paul, I guess, or bugging Paul. So do you guys look at the pore water mercury content at all? Have you thought about that, or are you?
Not yet. We've done it with carbon. We've done it with DOC, and I found very, very high concentrations of DOC in the pore water. So, you know, if you follow the relationships between organic carbon and mercury, we would expect to see the same thing, I think. It's something that's on our docket, you know, something to look at. We measure moisture content to see if there's some relations there. So far, not really anything sticking out at us. So yeah, I mean, you know, in order to get the pore water, you know, from a laboratory standpoint, that's going to be a little bit of a challenge, and you know, and I think we'll, you know, centrifuging and the other methods of extracting the pore water. And again, you got to keep it all mercury clean too. Mm-hmm. So you know, that's the big challenge there. So
yeah.
A question for Bob Gerlach. Bob, how far back does the fish monitoring program go in Alaska? And second part of that, if somebody was interested in correlating mercury measures in water or permafrost and locations that freshwater fish were taken from, would data be available for collaboration on that?
Yeah, it sure would. We started collecting samples in 2001, but what we've done is are working with our other collaborators to try to use a set of data quality standards so that we can take data that's, say, for example, at USGS or US Fish and Wildlife, and be able to include it in our database so we can go ahead and display it on our map, so that we can go ahead and have a more robust sampling or a data set for people to look at.
I have a question for Bob. Bob, it's good to see you again. I haven't seen you since the 2015 AFE, I think, right?
Yeah, that's it.
Yeah. So have there been any advancements in the understanding of how certain species of fish metabolize mercury into methylmercury? And, you know, I know there's been a lot of talk about the variability between species.
Yeah, I think it's a good point. We have not done a lot there, but from the little that I know about it, I think it has to do with their overall nutrition and diet. So we do see that, say, for example, a lot of our freshwater fish seem to have a higher concentration of percent methylmercury in their tissue than many of the marine species. And we do think that may be due to the selenium that is in the marine diet and how it would bind up and convert that mercury into a salt and keep it from the deposition there. The species difference, though, is really accentuated, I think, when we look at some of the marine mammals and some of the other terrestrial animals that we have just, we don't have much data in our database, but our other partners do.
Okay, good, thanks.
This is Jim McClellan again. One more, I promise I'll stop. This is, I guess, for Sarah and Bob. It looked like coverage on the North Slope is pretty minimal, and I'm wondering, is that just due to access, or is that less interest by the local communities? And just sort of to provide some context, I've been working out of Kaktovik and on Barter Island, and I know from talking to people there that that's the number one thing they keep bringing up to me as a concern is mercury and their health and wanting to know more about it. And that's not something that I do, so I would love to sort of point them in other directions. But anyways, yeah, just to answer my question, if you would, what's the scarcity of data on the North Slope due to?
Do you want to go first?
Sure, I can do that. I think the scarcity is more of just maybe the word isn't out there enough to the interested parties. I do think because our hair mercury biomonitoring is more of a convenience-based, perhaps we're just not seeing as many people from the North Slope in the types of meetings and conferences that were examples. If you do know of people would be interested, or if you know of a way to get the word out to the communities, we'd be happy to help that and get some more samples from that region.
Mm-hmm. Great.
And from our standpoint, I think it's the same thing. We do work with a number of communities that have had questions about mercury level and have helped with providing them some data. On the North Slope, it does seem to be difficult with the scattered populations, and we definitely have worked with a lot of the science programs in the schools, where when they end up having students come in during the summer for some summer programs, we've been working with some of the coordinators at those programs. So when the kids go out and collect fish or sediments, that they can send them down to the lab. And what we do is we put those on a priority basis so that we can get the data back to them within that 4 to 6 week time period, so they can actually have it as part of their curriculum for some of these summer programs.
Question for Sarah or Bob. Those fish consumption guidelines are a nice outgrowth of the programs that you have. I'm wondering if there's been any assessment of how well those are understood, or how well people comply with those guidelines.
Sarah, do you want to handle that?
Yeah, the short answer is no, but we have had some discussions over at DHHS about that question, and we did have an intern looking into that as well. So I can loop back to you on that with some more information.
And we did just last year on our webpage, Tom, set up a survey to ask people questions about their consumption of fish and what concerns they had, and help us with directing some of our outreach and data to try to help answer those questions. And we had it both from a survey for commercial fishermen as well as for consumers.
Great, thanks. Other questions from the group?
I actually have one more for Bob. Is there ever a concern with waterfowl in terms of mercury buildup? I guess it would be more in the more sort of carnivorous of the waterfowl species, but.
Yeah, I know that there have been a big concern with respect to the movement up the food chain to both even terrestrial species and birds, and I know there's been quite a bit of work done on the East Coast with respect to even evaluating mercury levels in birds and their behavior, both behavior for tending and nurturing the young, as well as reproductive capability, and also mating behaviors and even hunting behaviors to show how some of these elevations can go ahead and have some pretty significant impacts on their development and sustainability of populations that may be impacted by higher levels of mercury. We haven't done too much of that up with our program. It's basically we try to work with the university and some other collaborators to try to get them the data that we have so that they may be able to see those food chain impacts.
Yeah, I guess I was asking specifically about that as a pathway into back into local human populations. Again, from my experience in Kaktovik, in addition to eating a lot of fish, fish and marine mammals, a huge part of their diet is from hunting of waterfowl and consumption of waterfowl.
Yeah, I think we haven't seen much of an issue or concern with respect to the mercury, and it's been more of a focus on lead, actually.
Ah, interesting.
Can I just point out that there are quite a few comments in the chat box, so people can skim through those, and I'll add them to the meeting notes, but people can look at those.
Yeah, thanks, Sarah.
And somebody did just ask, what do you think about the, gosh, sorry, my screen is moving.
About the level of mercury
and glacial ice.
Yeah. Paul, do you see that question? Igor asked about, what do you think about the level of mercury in glacial ice?
We actually have another paper out that came out in, how long ago was that now? 2002, where we basically developed a paleo environmental record of atmospheric mercury deposition from an ice core in Wyoming, and again, there's a somewhat well-used figure in that publication that shows the deposition of mercury from about 300 years ago to present, and how we were able to tease out the different signals. We saw Krakatoa, we saw Tambora, we saw the gold rush of in the mid 1800s from California, when they were using mercury to amalgamize gold, and then they would burn it into the atmosphere. We saw all those signals, and along with that, we saw the constant increase in anthropogenic contribution from the baseline that we had well before the Industrial Revolution. So yeah, I think so. I haven't even actually seen the question yet. It was about ice cores. Yeah, I'm not finding that. Oh, there it is. Oh, and glacial ice. Yes. Yeah. Well, and so based on those numbers, you know, and we've only took one core, but there has been a lot of other work done and measuring mercury coming off of you know, the large sheets, you know, Greenland, and some estimates being made that, and also glaciers in southeast Alaska. There's some people that are starting to do work and looking at that, and seeing that could be quite a contribution to the global cycle that we should not ignore.
Okay, I see Igor send you his thanks through the chat box. Thanks for picking up on that.
Yep.
Paul, when we post your slides, perhaps we should make a link to your recent publication on this topic too.
Sure, I can provide that for you.
Okay. Good. Well, any other questions before we wrap up?